Okay. There's a slight change in title. The brain circuits I/O is replaced by volume EM platform because all these developments are quite recent, and also the thinking process on what actually the software stack that I'm going to present is. So volume EM is an image processing and analytics platform for large-scale electron microscopy connectomics. And first, I want to give you a short motivation of why to do EM connectomics, mapping brain circuits from using electron microscopy. So many things that also are, like in general, the topic of this workshop boil down to the von Neumann-Eckert architecture, where conceptually the central processing unit and the memories are separate things. And in principle, there's a bottleneck there, this transfer. And it's not only like a physical bottleneck, but also an intellectual one. And this permeates through all like more recent developments, also like parallelization, GPUs, and so on. It's still, the bottleneck is there. And the motivation for connectomics is essentially, at least like one motivation, is that the brain biology and evolution has come up with a processing architecture where memory and the processing units are combined. And connectomics is about discovering the connectivity, the architecture. So this is like a very high-level view on things. What exactly is connectomics doing? So it's about mapping brain circuits, and in the lab of Rainer Friedrich, we're concerned with zebrafish, in particular the olfactory system. And what one does there is to have a zebrafish brain and do a complicated process and produce these samples and image them with a microscope, an electron microscope. And in particular, we have a serial block phase electron microscope in this process. So what happens here is that you have the sample, and the knife cuts the surface, and images are taken in tiles by an electron beam, and you end up with the serial images. They're also tiled in 2D. So this is how one single image looks like, so really the raw data. I have this pointer here, I guess. I don't move around too much. Okay. So what you can see here are cells. These are cell outlines. The white space is the extracellular space, and you see stuff inside. And what we're trying to do is to first do a classification of these images. So you see different colors represent different things in these objects, like mitochondria, the cell interiors, or this vesicular structure inside the cells. So this is the first thing we want to do with this data set that we acquire. And the second step is segmentation. So we want to really figure out the single cells, these neurons, how they're connected to in 3D. And this is to give you background, basically now, how are we going about this? So the big data workflow and the challenges I'm going to present are segregated here in these three different broad areas: acquisition, image processing, analysis and visualization of these neural network data. And what you can notice here is also that this is not like a linear flow, but there are also these arrows back. And they're kind of like important, like from experience, that when you do image processing, you might also want to inform the acquisition process that maybe something is going wrong, and you should modify parameters or stop it. And also when a biologist analyzes the data, there should be some way to reprocess just these parts to improve on the results. So there's also a flow going back. And I'll briefly talk about a few of the challenges in these different areas. We've heard now the large data set sizes, terabyte to petabyte range in our particular case, or even exascale range that might be coming up in the next 10 years or so. Then you have to deal with millions of images, image files. They are located in 3D. And then also data acquisition rates are increasing as the microscope technology improves. So what we're currently dealing with are on the order of 3 terabytes a month with this SPAM method that is existing here at FMI. But there's also a possibility with other techniques like so-called multi-beam scanning electron microscope to acquire 2 terabytes per hour. So yeah, if you have a student and you want to acquire a data set and he has then hundreds of terabytes, then you have to, yeah, it's really limiting. So this regime here on the 3 terabytes per month, this is basically the infrastructure I'm going to present. That's for these data ranges. Then on the image processing side, one particular thing for the EM data is that this has a very high information content. So almost every pixel means something, and you want to know what it actually is. This might be different than in other topics where you have cells that are very sparse in the images. Then also the resulting data types are very heterogeneous. So it's the tiles, the raw tiles, the metadata. Then these images are deformed. You have deformation fields. When you segment them, you have 3D components, skeleton representations of these objects, or meshes, or semantic annotations of the cells. And you need to keep track of these and be able to work with them. Then there's also the demand for like volumetric random access in very large data sets by humans and computers. And the whole field is still kind of like in the process of method and algorithm development. So for alignment of the data or segmentation, this is not completed. So it's essentially starting the UI, to build the UI is a bit too early at this moment, mostly because algorithms and parameters might change. And then it's also, there's a need for diverse information IT experience, so high performance computing side, system administration, databases, GPUs, software engineering, and web frameworks. So it's a lot of things. And usually, like, not a single person can deal with all of them to a sufficiently expertise level to build these infrastructures. So this is one of the challenges as well. And on the analysis side and visualization side, this kind of like data that we're aiming to produce is the first of its kind, so to speak. So it's like this neuron embedded in 3D space, and they connect at synaptic locations, and there's not much of this data around, so it's hard to really know what you exactly want to analyze. So you need to customize your analysis data and visualization methods still. And also that many people might want to work on this data simultaneously and update and annotate it. So it's just like one big data set, and you have many users that want to access the same data set at the same time. So what would be the solution? What would be the solution that I'd like to present to you is on the acquisition side, it's a software tool called SpamImage that is developed by Benjamin Tietze in our lab, which is like a GUI acquisition software for the spam microscope. Then the volume EM platform sits in the middle and provides the processing layer orchestration or organization. And on the analysis and visualization side is a tool called CatMate. And I will just briefly give you a quick overview of this of SpamImage and CatMate, and then talk mainly about the volume EM infrastructure. So SpamImage, this is how a view looks like. So it's an acquisition platform for zero block phase imaging, as I said. Not yet publicly released, but will be soon. And if anybody's interested, contact Benjamin. So essentially, what you can do is you tile your images, and you have the option to have multiple tile grids that are overlapping. Then there's quality control monitoring during the acquisition for debris detection and so on. Things that you really want to do operating the acquisition process. And then on the analysis and visualization side, there is CatMate. It stands for Collaborative Annotation Toolkit for Massive Amounts of Image Data. This is the web view, so it's a client-server application where you essentially you see the raw images and you see the skeleton points and synapses, and you are able to like represent your data in different ways as networks or in 3D visualizations. And I think the thing is not playing here. It would be like a rotating thing. And you can work with the network, so you can select neurons, look at their connectivity, and do many kinds of things. And it's been already used in more than 20 publications, roughly, which speaks, I think, for the quality or the usefulness of the tool. And it has also an API to access it from Python or MATLAB to get data and push data back to the system. And the whole intention of this is also to enable people to work collaboratively. So they just open their web browser and access the data and then start to do it. And in the backend, it's a Postgres database who manages all the data. But now onto the Volume EM platform. This is just a brief, general overview with different areas, like the EM facility, the internal networks that exist on many institutions, then the cluster infrastructure or the internet, external collaborators. And essentially, so you have a data flow from the microscopy system to a workstation, backup system, and Dean and Enrico talked about this. And data is then pushed to the file server in the internal network and metadata to an application server from the Volume EM platform. And there are clients inside the institute who can access it, and also external clients. And in case you need to do really a lot of computation, there is a way to communicate with a high-performance computing infrastructure like at Novartis, the cluster at Novartis. So to go a bit more into detail, what actually the platform is, I made this diagram here to give a bit more inside view. So what you see here on the sides are essentially web APIs, RESTful APIs for different topics, and these serve as an interface to the application server and the database that stores data. And so you have one user here, like me, that works in a Jupyter notebook or in CatMates and accesses its data. Like the data is coming from the spam image through this acquisition interface, pushed into the server infrastructure here. And what it does, as just one use case, is that you can render on the fly the images that are coming in CatMate. So you can open your web browser and you can see the images coming in. They are placed roughly where the microscope thinks they are. And you can inspect the data through the web browser as the data comes in here. And at the same time, the server manages the tile metadata. This gets stored in a database inside here. And attached to it is the shared storage system. So we heard about this. This is the tungsten system here at FMI, like with RAID or SSDs, or even the archiving and the communication is the platform or the application server communicates with this as a storage for the files and at this database level with different types of databases. And there's also this task queue, which is a way to distribute tasks that come in. We saw this very nicely with the tissue maps. For instance, when people do things and they submit a job, there's kind of like a system to dispatch then the jobs to CPU or GPU resources. And important here to say is also that this, you can set up VMs, virtual machines, and add more resources as you need them in-house. And this can be done without any high-performance computing infrastructure. So this is a very nice way to just scale as you need in-house, let's say, and then if it's not sufficient, you can go to a bigger system if that's necessary. But I won't talk about this right now. And yes, so, okay, important here to say also that these different APIs, of course, things happen. So for instance, in terms of alignment, aligning the data, what you can do then is to, as data is acquired here, I can say, okay, now start the alignment process during the acquisition, for instance, already, and produce some results that I can look at and maybe notice error or so forth. So this can be done independently. So it's just a matter of data coming in and it's saying, okay, align this data, and then there's a task queue that performs the job and produces the results back here into the database, and you can look at them here. So there's many data flows, but the API is essential to basically interact with the whole system. And yeah, I think time is already almost up. Just as an overview, a summary, this platform gives you a RESTful web service API that is accessible via REST. And this is for data management and monitoring, or the data flow control. And then there's also, the system is built in a way that you can modularize analysis codes for classification and segmentation, for instance. And there's a scalable data processing aspect with a distributed task queue, and also this on-the-fly operations on all aspects, basically, of the whole process, which are quite important. And I didn't talk about this, so there's also some versioned alignment procedure that are implemented in the Volume EM platform. Mostly it's that at the moment. And yeah, so just an overview. So for the documentation, you can have them, for instance, for the acquisition API, you can say, okay, start a new acquisition run. And at the moment, SpamImage uses this API, just tell the platform, okay, I started a new acquisition, and it pushes the metadata into the system during acquisition. And there's also a lot of other functions in the other APIs to use. And yeah, I'll briefly show this image. I don't know why they don't show up, but I'd like to acknowledge Rainer Friedrich, with whom I've been working in the lab for a bit more than a year now, and also Christelle for the whole support for the EM aspects of things, and also Stephan and the whole IT team to get me running on the infrastructure side of things here. Thank you very much.
This is a very naive question, but I totally missed it. So I mean, this track